Federico, hi.
Hi, Julie.
You've got quite a bit of paperwork here.
Dr. DeSalle just stepped out. We're trying to go over the data that we've obtained during, I guess, the now 3 visits to your company.
Yes.
And each time we've learned something new and are finding additional applications to the to this unique spectrophotometer that you have, the Clarity.
Yes.
The first advance was we repeated. This is a paper from George Wald's classic paper, The Molar Extinction of Rhodopsin from 1953, and I don't know if you can see it. There's
only 51 years ago.
A spectrum of rhodopsin.
Mm-hmm.
After rhodopsin is bleached by light, you see a drop in this particular peak. Of course, this is purified protein.
Okay.
And what we've been able to do with your system is a similar type of a measurement with the very crude preparation of so-called wrought outer segments from bovine retina.
Okay.
This is very opaque. You can't even see through. It looks kind of milky.
All right.
But the Clarity was immune to the scatter and has allowed us to obtain a rhodopsin spectrum. This is the rhodopsin here, and
Fantastic.
this is after bleaching.
Right.
So then on the second visit, we said, well, what about if we use living cells? Can it work? And we obtained a similar kind of result. These are the cells, and these are individual scans from the rapid scanning monochromator in the Clarity
1000.
Was it the 1000?
Yeah, the Clarity 1000.
In the dispersion cell.
Right.
And then this is, of course, this is going down, and this is going up. So the scatter of actually bulk cells doesn't interfere with the obtaining of the spectrum. So that opens the door to being able to do physiological experiments, allowing us to use living, breathing, ticking cells.
Fantastic.
And then on this visit, we tried a different application. We have an interest in studying how vitamin A degrades, and the protein that we're studying is a high molecular weight lipoprotein in the extracellular matrix around the rods and cones, and we think that this protein protects vitamin A from photodegradation, and we have a paper now published in 2014 in experimental eye research that shows the protein has this function. But we need a more sophisticated way to deliver light to vitamin A and follow its degradation in vitro. And that way we can test our recombinant proteins to get a better handle on what the mechanism is for IRBP's remarkable ability to protect vitamin A from photodecomposition. So here we're taking advantage of the unique chamber that you have, which is a cuvette shaped like a sphere. And this allows us to put the actinic or damaging light 360 degrees around the sample, hit it with light, and do that between scans. So we do a rapid scan, hit it with ultraviolet light, do another scan, and this way we can follow very accurately and characterize the kinetics of the photodegradation. Then using this system, we'll then introduce the vitamin A binding protein and characterize hopefully the mechanism of how it protects vitamin A from degradation. But we first need to characterize how vitamin A degrades and in what ways before we can figure out, or at least we can figure out in parallel with the role of the protein in protecting the vitamin A, if that made sense.
Okay.
But, so with that turned out the dispersion cell, which I think you call the, the DSPC for...
DeSalle suspension presentation cavity.
Yeah, essentially it's an integrating sphere, cavity, where the light comes from 360 degrees. You have a kind of a gas of light, essentially.
Exactly.
Because in a standard cuvette, the light goes through the cuvette, and for the purpose of this particular project, a lot of the vitamin A could hide from the beam. So you don't know whether the whole sample is receiving the light, you see.
Right.
So the advantage of the integrating sphere is that the whole
Into the
samples
cavity.
receiving the light
Yes.
in the cavity, and then we can very carefully not only hit the cavity with light of defined wavelength and intensity, even making use of LEDs,
Right.
and at the same time follow the degradation of the vitamin A by rapid scanning.
Yes.
And then using the kinetics packages, we've been able to actually find evidence for several intermediates during the degradation of the vitamin A. So it's a different application.
Yes.
It doesn't utilize the anti-scattering effect of the or the immunity to scattering that the
Nor
clarity
the
has.
long path length. That's not an advantage for you in this case either.
Well,
but having
yeah, but that does increase the sensitivity tremendously. Yeah,
that's true.
so that's quite. But of course, the fact that the instrument is on an optical bench allows you to do different things and experiment with different paradigms and combinations. So it's been a very productive 3 visits, and we're hoping that we submitted an abstract to our society meeting, which is called ARVO, Association for Research in Vision and Ophthalmology, in May. Hopefully the abstract will get accepted and we'll present some of this data formally at that meeting. But thanks for letting me come over and hang out in your laboratories.
Well, it's been a joy having you, and as we like to say, get to work now and get this published. I think you've got a lot of work ahead of you, and you've already got 1 manuscript in preparation, and I'm sure the others will fall out quite quickly and naturally too.
Yeah, I think so. I think we're going to be having some nice things to say.
Cool. Well, thank you very much,
Okay.
Federico.
Okay, thank you, Julie.
You're welcome.